And I'm very honored today to introduce our keynote speaker, Professor Virginijus Siksnys. Hopefully my Lithuanian was good. And Virginijus is a leading European scientist and a pioneer of the CRISPR-Cas9 field. He holds the position of distinguished professor at Vilnius University in Lithuania and serves as the chairman of the board of the Life Center of Life Science Center in Lithuania. As you all know, his groundbreaking work on CRISPR-Cas9, and in particular on biochemistry of CRISPR-Cas9, paved the way to all the innovative application that we are going to hear during the next few days. In recognition of his seminal contribution, Professor Siksnys has been honored with prestigious awards such as the Warren Alpert Foundation Prize and the Kavili Prize in Nanoscience. And I can see him ready, so please welcome me in introduce.
Thank you. So I don't need it. Marcello, thank you very much for a kind introduction, and it's my great pleasure to be here and see so many people here. So Karen told me that there are around 350 people here at the meeting, and all of you are interested in biomedical applications of CRISPR. And actually, this brings me back nearly 18 years ago when we had a first meeting in Berkeley at Stanley Hall. And it was first meeting where people actually came to discuss a new antiviral defense system that was described reported in Science paper. And these 30 people who came to the meeting, they came from different parts of the world, and including Francis Mojica from Spain, Rudolf Barangu, Philippe Horvath, Eugene Koonin, and others. And all of them were actually interested in this CRISPR new antiviral defense system that was called CRISPR. But ironically, at the meeting, actually, most of the people were talking about their previous research experiences. Few of them were talking about CRISPR, but no one was talking about biomedical applications. And today, actually, the meeting is focused on biomedical applications. So I thought that probably somebody has still talk about CRISPR. And the CRISPR is actually antiviral defense system in bacteria, and it functions as an adaptive immune system. When viruses, phages infect bacteria, so most of the bacteria die. However, small number of bacteria survive, and it turned out that survivors were able to hijack small pieces of invading viral DNA and insert them into a so-called CRISPR region as spaces, and then bacteria can use these spaces that came from phages as templates to make small RNA molecules that, together with Cas proteins that are encoded by Cas genes that are located nearby of the CRISPR array, assemble in a so-called effector complex, and then this effector complex is able to recognize invading phage during next round of infection and destroy it. So in this case, CRISPR-Cas system really functions as a very primitive adaptive immune system of bacteria because it's able to memorize the invader by inserting piece of invader's DNA into the CRISPR array and then later use this information to recognize the invader. So in my lab, we were interested in structural and molecular mechanisms of CRISPR systems, and we focused from the very beginning on the CRISPR-Cas systems that were present in Streptococcus thermophilus bacteria, and this is an industrially important bacteria because it's widely used for making cheese and yogurt. And we got this bacteria from Philippe Horvath, who was actually the first author of the science paper, and started to look at the CRISPR systems that were present in this bacteria. And in fact, there were 4 different CRISPR systems in a specific bacterial bacteria, and actually they are called CRISPR 1, 2, 3, and 4 at that time. And of course, the question was actually which one you would like to choose for your studies, because you don't know anything about them. And we decided to focus on a CRISPR-3 system of Strep thermophilus because of the 2 reasons. First, actually, it contained the smallest number of genes, only 4 genes in the CRISPR locus. And then, actually, one of the gene, the Cas9 gene, encoded a protein that actually showed the signature motif that we previously seen in restriction enzymes. So that was a good hint that that's kind of, this could be a protein that could have a kind of nuclease activity. And we didn't know how to handle the Strep thermophilus in the lab, so we decided to try to move the CRISPR-Cas system from Strep thermophilus into E. coli. And to our surprise, this experiment worked really well. So we were able to transplant the CRISPR system from Strep thermophilus and put it in E. coli. And this CRISPR system, in fact, was functional because it provided interference against invading phages or plasmids if a matching spacer was present in the CRISPR array. And then using genetic tools that are easily available for E. coli, we were able to show that actually Cas9 is the sole gene that is required for interference. And this was, in fact, the first demonstration that CRISPR-Cas systems are compact transportable units that could be transported between different organisms. And then the next obvious step was just to try to isolate the Cas9 protein, and Gedrius Gasiūnas, who was a PhD student at that time in the lab, and he's present in the meeting, actually isolated the Cas9 protein, and it turned out that Cas9 actually purified these 2 RNA molecules, CRISPR RNA and tracer RNA. And then using a set of biochemical experiments, we were able to show that actually Cas9 complex with RNA functions as RNA-guided desoxyribonuclease. So using CRISPR RNA, it binds to the one complementary DNA strand, making a structure that is called R-loop. And then Cas9 proteins, using 2 different active sites, introduce double-strand break into the DNA. So the simple modular organization of Cas9 ribonucleoprotein complex, where specificity is encoded by CRISPR RNA and cleavage is provided by Cas9 protein, paved the way for engineering of universal DNA endonucleases. Indeed, by simply changing CRISPR RNA in the complex, one can target Cas9 nearly to any site in the genome, and then Cas9 will introduce a double-strand break, and this double-strand break trigger cellular mechanism of repair that could be harnessed for gene knockouts or knock-ins. And then afterwards, then Cas9 actually became a universal genome editing tool that was applied in very different model organisms. And if you look at the CRISPR timeline, so the BC, it means before Cas9, the CRISPR-Cas system was just a peculiar antiviral defense system, but in 2012, actually, it was a turning point. So in 2012, the Cas9 became a genome editing tool. And then, after Cas9, everyone is now talking about gene editing technologies. But actually, this just shows you that the line between basic research and technologies is very thin. One day it could be a basic research carried on in order to understand and answer very basic biological question. Next day it could become a technology. And you already heard from Jimmy about the power of Cas9 technology and how Cas9 technology could be used to treat genetic diseases, because it, in principle, allows to reverse mutations. So the Cas9 is a really great tool, and it's already in the clinic, and there are many other clinical trials. But of course, there are still challenges that are related to targeting space limitation, delivery of Cas9 into the cells, off-target cleavage, and others. And of course, one should think about at least two different solutions how to overcome these challenges. And one of them is one can take an existing Cas9 and aspergillus Cas9 and try to engineer the existing Cas9 in order to overcome these limitations. And another approach is just to dive into natural diversity of CRISPR system. In my lab, actually, we have focused on this natural diversity approach. And the CRISPR-Cas systems are really very diverse, and they are broadly divided in two classes, class 1 and class 2, mainly, and the major difference is in the structure and composition of effector proteins. In the class 1 systems, the effector protein is assembled from multiple subunits of the protein that bind CRISPR RNA, and this is exemplified by cascade complex of type 1 CRISPR-Cas system. In the class 2 systems, the class 2 systems rely on the effector that actually is a single protein like Cas9 or Cas12, and this single protein binds single or two RNA molecules. And the effectors that belong to the class 2 are also very diverse, and they are further subdivided into different subtypes like type 2, that is exemplified by Cas9, type 5 that's exemplified by Cas12, and Cas9 and Cas12, they both act on the DNA, while actually the effectors that belong to the subtype 6, as exemplified by Cas13, they act on the RNA. And I already mentioned that limitation of targeting space is one of the challenges when Cas9 is used as a genome editing tool. And the specificity of the Cas9 is dictated by the CRISPR RNA that base pairs with matching DNA strand through the Watson-Crick interactions and makes a structure that is called R-loop. And in order to initiate the formation of R-loop, the Cas9 protein has to recognize a short nucleotide sequence that is called PAM, or protospace adjacent motif, in the vicinity of the target site. And the PAM sequences are recognized by Cas9 protein, and different Cas9 proteins show different PAM requirements. And this PAM requirement, in fact, limits target site selection. And for example, for aspergillus Cas9 that recognizes GGPAM, it constrains targeting to every 8 base pairs in a human genome, making nearly 75% of mutations that are associated with human disease non-accessible for Cas9 cleavage, if a very precise cleavage is considered. So we thought that actually we can probably overcome this PAM sequence limitation of targeting space looking at the space of Cas9 orthologs. And we developed a phylogeny-guided bioinformatics approach that allowed us to identify thousands and thousands of different Cas9 variants. And then actually we grouped these Cas9 variants, building a phylogenetic tree that contained at least 10 different sequence clusters. And then we selected 100 of Cas9 proteins that belong to different sequence clusters and actually aimed to analyze biochemical cleavage requirements of these Cas9 proteins. And for each new Cas9 protein, 2 requirements has to be established. It's a guide RNA sequence and then PAM requirement. So the guide RNA sequence could be predicted bioinformatically, but actually for the PAM sequence, one need to develop an experimental method. And actually we developed a pipeline for PAM characterization using in vitro transcription translation system. And in brief, Cas9 proteins were actually produced in the IVT system, and then Cas9 RNPs that were assembled in solution were used to cut a DNA library that contained a single target site that was flanked by 7 nucleotides in the vicinity of the target, and this position corresponded to the PAM sequence. And then actually we analyzed cleavage products using deep sequencing and identify PAM sequences in the library that license Cas9 cleavage. And this simple screen actually reported a really huge diversity of the PAM sequences for different Cas9 variants. So we were able to identify PAM sequences, T-rich, C-rich, G-rich, and mixed sequences containing mixed nucleotides that were from 2 to 5 nucleotides in length. So the Cas9 protein, in fact, is a large protein that is around 1,300 amino acid residues in length. And when people think about human therapeutic applications, so adeno-associated viruses or AAVs are preferable vector for delivery of gene editing tools. But AAVs have their own cargo packaging limitation, and therefore it's difficult to package into a single AAV a large Cas9 protein and guide RNA that is required for targeting. So therefore, there is a considerable interest in smaller Cas9s or other genome editing tools that would be compatible with AAVs. And in order to address this challenge, actually, we again looked at the natural diversity of CRISPR-Cas systems. And the Cas12 nuclease family actually is part of the type 5 CRISPR-Cas systems, and it's very diverse. It contains many subfamilies and different proteins, and all of them actually contain a single RAFC active site, but they actually differ very much in length. So the shortest Cas12 family members that belong to the Cas12F subtype are just around 400, 500 amino acid residues in length, while actually the first Cas12 family enzyme, Cas12A, is as large nearly as Cas9. And there are actually variants with size in between the Cas12F and Cas12A. And therefore, this diversity actually is a really good resource to look for novel Cas12 enzymes. And a few years ago, Schmackhoff and Harrington, they reported a new Cas12F family of proteins that is part of type 5 CRISPR-Cas system. And actually, it formed quite a diverse family, and they fall into at least three different sequence clusters. And we decided to look whether these small Cas12 family nucleases that are around 400 or 700 amino acid residues in length, in fact, are able to cleave double-stranded DNA, because in the initial paper on the characterization of Cas12F family enzymes, it was kind of reported that it cuts only single-stranded DNA. And to address this question, we actually used the same assay, biochemical assay, based on the cleavage of plasmid DNA library that I described you when I talked about our studies of Cas9 orthologs. And we used this assay actually to look at the biochemical cleavage requirements of different Cas12F variants, and we selected 10 different Cas12F variants. And what we found that actually they all actually were able to cleave DNA, introducing a double-strand break, and they all were dependent on the PAM sequence. And the PAM sequence actually usually was T-rich, but there were also sequences that showed actually CC requirement for the DNA cleavage. So then we decided to select 2 proteins that recognize different PAMs, and then to look whether these proteins are in fact able to introduce indels in human DNA. And we expressed these 2 proteins in a plasmid, and then actually transfected hex cells and looked at the indels after 72 hours. And what we found that indeed that actually these proteins showed very, very weak indel activity in human cells, but it was promising that at least they were not dead. So then we asked the question, is it possible to improve their editing activity? And Josh Young at Corteva developed a yeast-based screen. And in this yeast-based screen, he generated a protein library and transformed into a yeast actually the guide RNA in the library actually was targeting the AD2 gene. And if the nuclease is able to cleave the AD2 gene, so then the yeast turn red. And this was a kind of marker that showed that there is a nuclease activity. And if you look at the wild-type protein, there are nearly no red colonies. So you see some red colonies at 45 degrees Celsius. And the reason is that, in fact, the thermal profile of the Cas12F nuclease that we were investigating, in fact, is shifted to higher temperatures, to 50 to 60 degrees Celsius. So therefore, at 37, it's barely no cleavage activity. However, when he analyzed the library, he found that actually in the library, there were variants that showed activity at 37 degrees, and then actually when he combined these different variants, he found that actually nearly all colonies turned red at 37 degrees Celsius. And in vitro studies revealed that actually these mutations that were in the library, they shifted thermal profile of the Cas12F family nuclease. And then the obvious question was whether these mutations that improved activity in yeast translate into human cells, and the short answer is yes, because actually you can see that the wild-type Cas12F shows very low cleavage activity. However, the mutant variants showed really increased cleavage activity. And when actually Josh engineered also the guide RNA, so the cleavage activity increased to really impressive numbers. So this simply shows that actually protein guide RNA improvements that were actually obtained during the screen of yeast library actually translates into human cells and results in really high increase of indels. So it looks like the Cas12F as a promising genome editing tool But of course, it recognizes quite a long PAM sequence. And this quite long PAM sequence that is DDTC actually limits targeting space. And if you look and compare it with Cas9, so that's kind of its really considerable limitation. So we thought, okay, maybe we can change the PAM sequence of Cas12F protein. And in order to do that, we decided to solve the crystal structure of Cas12F nuclease. And we did it using cryo-electron microscopy. And the structure revealed that the protein is arranged of two lobes that are characteristic for Cas12 family nucleases, and the RNA is sandwiched, guide RNA is sandwiched between these two lobes. And then actually, looking at the structure, we were able to identify amino acid residues that are responsible for the PAM recognition. And then actually we changed some of these residues guided by structure. And then what we found that indeed one of the changes introduced at lysin 85 expanded targeting range and changed PAM into YTC. And this actually change of the PAM expanded the targeting space. So far I just described you about the smallest miniature Cas12 family nucleases. And then in the next part, I would like to talk about the Cas12L nuclease that has a size in between the Cas12F and Cas12A. And actually, the Cas12L nuclease was isolated from the environmental metagenomic samples, and it forms a distinct type of type 5 CRISPR-Cas systems, and its size is around 860, and RNA that is bound to it is around 170 nucleotides. And so Gedros and his team at Kazim, they were able to show that Cas12L cleaves double-stranded DNA, and it requires CCY PAM, and it produces 5 to 9 nucleotide overhangs in the DNA after the cleavage. So in order to understand actually the structure of Cas12 protein and also use it as a template for further engineering of it, we solved the structure of Cas12L protein, and it revealed a kind of unique structure. It was a protein monomer that was sitting on the large RNA like a bracelet, and this large RNA made two different arms that actually function as a scaffold for effector protein assembly. And this structure was different from other proteins of Cas12 family because it contained a rough C domain that was a rough C lobe that was more or less similar to other Cas12 nucleases, but N-terminal lobe was very different. And we used this structure actually to improve the activity of the Cas12L protein. So initially, Cas12L protein showed really weak activity in human cells. However, guided by the structure, Gedros and his team, they were able actually rationally redesign the Cas12L protein and significantly increase its DNA editing activity in human cells. And then actually when he used large language models to further improve the Cas12L, actually he really achieved remarkable results that now Cas12L was able to cleave most of the targets with really respectful cleavage activity. This simply shows that if you look at these nucleases that are present in the CRISPR systems and they show a really low activity, so simply by doing some kind of engineering of these nucleases, you can really significantly increase their editing capacity in human cells. So, and then Gedros also was able to show that Cas12L could be delivered into human cells either through the RNP nucleofiction or mRNA transfection. So some of the data were missing, sorry about that. So, and if you look at the PAM of the Cas12L nuclease, actually it's a CCY PAM, and it's located at the 5' end of the target. So the Cas9 protein recognizes GG-PAM, but actually it's located in the opposite DNA end. So therefore, actually one can use the same target sequence to benchmark Cas12L and Cas9. And this benchmarking exercise actually showed that in most of the cases, I don't know why the data is missing here, but I'm not hiding anything. I just want to highlight that in most of the cases, the Cas12L nuclease had an activity that was comparable to Cas9, SP-Cas9 on most of the sites. So actually, my intermediate take-home message is that I showed you that Cas9 orthologs are a rich resource of potential novel genome editing tools, and Cas12L nuclease shows a really remarkable DNA cleavage activity similar to Cas9 in human cells, and engineered Cas12F miniature nucleases are also active in eukaryotic cells. And of course, the interesting question that remained in the field was actually where these Cas12 nucleases that belong to this large Cas12 nuclease family came from. And if you look at the phylogenetic tree, so the closest neighbor of miniature nucleases is called TNPB. And TNPB actually has nothing to do with CRISPR system. It shares the same RFC active site with Cas12 family nucleases, but there is no CRISPR array in the vicinity. So TNPB, in fact, is a part of mobile genetic elements that belong to a specific insertion sequence 200, 605, 607 transposon family. And these mobile genetic elements move between different locations in the DNA. And these transposons, in particular of the IS200 family, they are probably most ancient and simple transposons. They usually are comprised, or often they are comprised of two genes, TNPA and TNPB, that are flanked by left and right end transposon sequences, and also requires a specific TTGAT sequence for this Deinococcus radiodurans transposon as a site where excision or transposon excision occurs or transposon is inserted in different location. And this transposon, actually the role of TNPA protein encoded by TNPA gene was well known, actually. The TNPA is a transposase that enables transposon mobility between different sites. And the transposon actually is excised from the DNA during replication, and it's excised as a single-stranded DNA intermediate from the lagging DNA strand. So the role of TNPA was clear, but actually what TNPB is doing in this transposon was still the question that has to be answered. And we decided to isolate the TNPB protein from Deinococcus TNPB that is encoded in Deinococcus radiodurans transposon, and Gytis Druteika and Tautvydas Karvelis, they actually expressed TNPB protein in E. coli cells and were able to isolate TNPB protein. And it turned out that TNPB protein purified from the cells together with RNA molecule, and RNA-seq revealed that this RNA molecule was around 150 nucleotides in length, And it corresponded to the right transposon sequence that is shown in blue. And also, it corresponded to the part of the RNA that actually encoded that transposon. And then at the end, it had 16 nucleotides that originated from the plasmid DNA. Since this RNA actually contained the right transposon element, we called this RNA rRNA. And then, of course, the question was whether the TNPB that is bound to the RNA functions as RNA-guided nuclease. And to address this question, we used, again, the same assay that we developed for Cas9 proteins and Cas12 proteins. We just expressed TNPB together with rRNA targeting a single site in the plasmid library, and then looked at the biochemical cleavage requirements for double-strand break. And what we found that this TNPB protein was able to cut DNA, introducing a double-strand break, but it required a PAM sequence that is TTGAT. And if you look at the PAM sequence that licensed the cleavage of the DNA, you can see that it overlaps with the TTGAT sequence that is required for transposon mobility. So therefore, we called this sequence TAM, or transposon-associated motif, in parallel to PAM, or protospace adjacent motif. So taken together, these data show that, in fact, TNPB functions as an RNA-guided DNA endonuclease, and the target site of TNPB is composite. It's comprised of TAM sequence that is recognized by TNPB, and then guide RNA and DNA sequence that is matching the guide RNA in the TNPB complex. So when the transposon is present in the DNA, this target site for TNPB is interspaced by inserted transposon. But when transposon is excised during DNA replication from the lagging strand, so then the TAM and sequence recognized by guide RNA come together, and they form a TNPB cleavage site. So, and then TNPB is able to cleave at this site, introducing a double-strand break. And then this allowed us to propose the role of TNPB in transposition. So what happens here, when transposon is excised from the lagging DNA strand during replication, so then two different DNA alleles are generated. So one allele actually still contains the transposon, and this allele originated from the leading DNA strand, and another allele actually that originated from lagging DNA strand lags the transposon. So then, in this alleles that transposon was excised, the target site is formed, and then the TNPB protein is able to cleave this DNA, introducing double-strand break, and trigger homologous recombination, resetting the transposon into the same position in the DNA. So, of course, the next question that was obvious was whether TNPB is able to cut DNA in human cells, and in fact, we were able to show that TNPB functions as RNA-guided DNA endonuclease that is able to introduce indels in human cells. And next, in order to look at how the TNPB RNA complex structure looks like, we solved the cryome structure of TNPB guide RNA complex together with DNA, and in fact, it showed a fold that was characteristic to the Cas12 nuclease family. It contained rectal lobe and NUC lobe, and RNA was positioned between these two lobes. And then, actually, what we did, we compared the structure of TNPB with other available structures of Cas12 nuclease family, and there are many of them. And it turned out that TNPB was most similar to the miniature nucleases of Cas12 family, and Cas12A actually was a most distant member of the family. When you compare the TNPB and the closest neighbor, Cas12F nuclease, and look at the RNA structures, so the RNA structures look really similar, but there is very important difference in the protein structure. So if you compare TNPB with Cas12F, you see that there are some extra decorations in Cas12F nuclease. And this is because Cas12F nuclease is a dimer, and TNPB is a monomer. And if you compare TNPB with Cas12A, so you see that actually there are a number of different decorations in the protein core. The protein increased, but the RNA size clearly decreased. And what we did next, Czeslavas Venslovas and his team, they actually compared the TNPB sequences that are available in databases, and it's really amazing. There are more than 1 million of TNPB sequences in different databases, and it looks like actually TNPB proteins make a really huge space, and TNPB proteins actually cluster into a single large universe where the TNPB that we characterize sits nearly in the middle, and then you can see that Cas12F proteins and other Cas12 family members, they are much smaller in numbers and form actually different universes. But this shows that probably TNPB was evolutionary predecessor of the Cas12 nuclease family. And the TNPB, in fact, is part of very different insertion sequence families, and it's in the insertion sequence families 605 and 607. Actually, TNPB is usually accompanied by serine or tyrosine nucleases that are actually performing the transposition. But there are also other families of this transposable element. For example, in IS1341, there is no transposase, but there is a TNPB gene flanked by left and right transposon elements. And this assumes that probably it's non-autonomous transposable elements, that TNPA is somewhere in the genome. So Gytis Druteika and Tautvydas Karvelis decided to look at the DNA cleavage requirements of these different TNPB proteins. They selected TNPB proteins that belong to very different families. And actually, using the same assays that I described, they were able to show that indeed these TNPBs are able to cleave DNA introducing double-strand break, and similar to Cas12 family nucleases, they all require TAM sequence that is different for different TNPB variants. Often they are T-rich, but sometimes they are also G-rich. And yeah, to finish my talk, I have just a few slides. And when I talked about the CRISPR-Cas systems, so they provided interference against invading phage DNA by cleaving DNA. And cleaving invading DNA is a strategy that many antiviral defense systems employ, including restriction modification systems. But of course, the question is, are there any other defense strategies or targets that are used for the antiviral defense? And Rotem Sorek and his team a few years ago described the so-called TORIS bacterial antiviral defense system. And it's comprised of two genes, THAS-A and THS-B. And THAS-A is comprised of SIR2 domain and SLOG domain, and THS-B is similar to eukaryotic TIR proteins. And Rotem and his team, they proposed a following mechanism for antiviral defense systems for tortoise system. So when phage DNA comes in, actually THSB protein, probably using the mechanism that still needs to be clarified, senses the infection, and probably this is achieved through the oligomerization of THSB protein. And then THSB protein actually synthesizes a cyclic ADP molecule using a NAD as a precursor. And this cyclic ADP molecule, in fact, acts as a signaling molecule that is bound by THSA protein. And then, actually, it triggers degradation of NAD and leads to the cell death. So this was the mechanism, but it was not clear how this THSA protein is getting activated. And it contains a CIR2 domain that is actually similar to NAD dehydrolases. So it could be that actually the CIR domain becomes somehow activated when actually this signaling molecule is bound to THSA. And then to address this question, Gedre Tamulaitinen in the lab solved a THS structure using cryo-EM microscopy. And what she found that actually in the presence of this signaling molecule, THSA forms a long helical arrangements. And these helices are actually formed from THSA tetramers. And each THS tetramer, it contains CIR domain and slug domain, and CIR domains are actually in the middle of the tetramer and slug domains are outside the tetramer. And these slug domains actually are sites where this cyclic signaling molecule is bound. And this binding of cyclic molecule actually triggers then oligomerization of these tetramers into a stack, a helical stack. And then actually this helical stack in fact results in the activation of NAD hydrolase. Looking at the structure, you can identify actually contacts between different tetramers in the stack. And if you mutate these residues in the interface, actually the filament is disrupted, as you can see from scattering experiments. But this disruption of filament also results in the elimination of NAD hydrolase activity. So these biochemical experiments demonstrated that this actually filament formation is actually an important step in the activation of the tortoise system. So taken together, these experiments allow us to propose an expansion or extension of the mechanism. So when phage comes in, actually THSB synthesizes the cyclic molecule that acts as a signaling molecule. It binds to the slug domains of THSA and then triggers polymerization, and this polymerization activates NAD hydrolase activity, and the system depletes NAD in a cell, and the cell dies. So, and right now, there is a conference going in Paris that people actually are discussing new antiviral defense systems, and there are hundreds of them actually described now bioinformatically. And this antiviral defense system, they use very different strategies to cope with viral infections. Some of them, as I described for tortoise, they use NAD degradation. Some of them actually depletes NTP pool. Some of them actually make holes in membranes. And so, as I said, hundreds of them are discovered, but why they are important? And if you look back at the history, so in 1960s and 1980s, discovery of restriction modification system resulted in recombinant DNA technology and was acknowledged by Nobel Prize. So in 2010 and 2020, CRISPR-Cas discovery of CRISPR-Cas systems resulted in genome editing technologies and also was acknowledged by a Nobel Prize. And now hundreds of them are identified. So the question is, what awaits us there? And of course, it's difficult to say, but what I suspect, very different chemistries, very interesting enzymes, and probably new Nobel Prizes, discoveries, and technologies. Thank you very much.
Thank you so much. Beautiful talk. Question for Virgis. Let's start here.
That was fascinating. Thank you. In the IS-2 system, how much of the target sequence has to be in the guide RNA in order for that to specifically insert at a new site?
Yeah, so the minimal requirement is 16 nucleotides.
60?
16.
16.
1/6. Yeah, it's comparable to CRISPR systems. Yeah, so.
Somebody else, yeah.
Great talk, thank you very much. Do you believe that also human cells possess something like the CRISPR-Cas system, maybe with other enzymes or another strategy?
You know, if you look at human cells, RNAi, it's analogous system to CRISPR systems because it uses RNAi or organoid systems that are used for silencing of RNA. Use the same principle, but actually there are no clues that these systems are evolutionarily related. However, if you look at these newly discovered systems like TORIS, antiviral defense systems that use different strategies, people actually speculate that these systems are actually similar to some of the elements of immune systems that are present in eukaryotic cells. And maybe actually it was kind of evolution from bacteria to these systems in human cells.
Hi, thank you so much. For the Cas12 engineering that your team did, I was wondering roughly how many substitutions were included on Cas12F, and then also how many, or what types of changes were done to the guide.
Yeah, so for the first part of your question, for Cas12L, you were asking specifically. F. Yeah, so it was, I would say it was up to 10 substitutions that occurred in very different parts of the structure that would be probably really difficult to predict rationally looking at the structure. So you need to go through this kind of directed evolution experiments.
Yeah, this one here.
Yeah.
Oh yeah, thanks. It's really fascinating. So I actually have two basic questions. You demonstrated performing the Cas12F and also L perform equally good with the benchmark with the SP-Cas9. So how well now it started about the off targets, and then the second one is actually most of us are still using the SP-Cas9. Typically, when you use it for therapeutics later on, the freedom to operate it is very, very limited, complicated. And what is the scenario for the Cas12?
Okay, regarding off targets, so I would say that both Cas12F and Cas12L actually, and the improved variants, they show no actually increased off-target activity on the predicted off-target sites. And actually, we're now looking into the detail into other sites throughout the genome. And yeah, regarding to freedom to operate, I will probably direct this question to Giedrius, who is present here and represents Kazime at the meeting. Yeah.
Yeah, here. Thank you for the amazing talk. I'm just going to ask kind of a little bit, maybe more on the basic science side. So why did these enzymes grow in size? Like going from, obviously, the transposons all the way to Cas9s and so on.
It's a good question, but probably there is no clear answer. But people actually also speculated that there is a weak correlation between the size of the RNA and protein size, and that actually, in the case of Cas12a, the RNA is small, the protein is big. In the case of Cas12f, actually is other way around. RNA is big, the protein is rather small. But it looks that during evolution, some RNA functions were overtaken by protein, and that resulted in expansion of Cas12 protein. But this was a speculation, so there is no clear evidence about that. Yeah.
Hi. Oh, sorry if I was first.
Yep.
Okay.
Sorry, from my side, I'm very curious about the Cas12f guide RNA actually, because my understanding, I also try to work with that. The scaffold is much more complicated what you receive in a SPCAS9, for example. How you can actually predict the structure, or how much you have to play around until you actually get the structure right, because there is a lot of loops, some of them may actually be not needed and stuff like this.
Yeah, you know, it's now quite easy using AlphaFold to predict protein structures, but it's still a challenge to predict RNA structures. So usually you have to address this question experimentally. You should go through deletions or substitutions in RNA just to see which parts are important or which are not. So.
Thank you. Great presentation. I'm here. Okay, my question is based on towards the end of the presentation, you told us that the future is looking towards the NAD degradation and the possible membrane disruption. I want to ask, has it been determined that for example, the NAD degradation, that there is a guided sequence that controls it, like we have in the Cas system, and also to also determine the possibility of also having off-target effects of the NAD degradation?
Yeah, I'm not aware if there is any RNA-guided NAD degradation system, because actually in this case that I described, actually the sensing of virus infection is very different. It's not based on the complementarity between guide RNA and matching sequences in the DNA, but probably there are other mechanisms how bacterial cells can sense virus. For example, they can recognize some of the key viral proteins, and then these key viral proteins can act as a trigger that triggers activation of the antiviral defense system.
I have a couple of questions as well. The first one, I'm surprised Alex, my colleague, didn't ask. So SPCAS9 is a very powerful enzyme, but most of the Cas9, also Cas12, this seems to be not so active, not only in human cells, but also biochemically. Do you know why? What is, there is any evolutionary force against that?
No, it's a good question, but still we don't have a clear answer. So initially when we started this exercise, we thought, okay, we'll have a bunch of new Cas9 that recognize different PAM sequences. But at the end, actually, nearly all of them worked well in vitro. In biochemical experiments, when you purify this Cas9, this guide RNA, it cleaves DNA and introduce double-strand break. But when you put these different Cas9s into cellular environment in human cells, actually things change. Not many of them are actually active in human cells, and we do not have a clear answer yet. There are some of them that are active, and we worked on some of them, and we're able to improve their genome editing activity, but in every case, probably you need an extra effort.
And have you evolved also TMPB, as you show with Cas12F?
Yeah. It's a similar story with TNPB. So you know that SPCAS9, actually, it was discovered in parallel to our Streptomophilus Cas9. And SPCAS9 actually is a perfect genome editor. Of course, people had to improve it. They did a lot of experiments to improve it. But the same story is with TNPB. So people now actually use different TNPBs in human cells to look whether they are good editors. And it turns out that the TNPB from Deinococcus rerodurans I described actually is the best editor as far as I know in human cells. And others behave rather poorly. But again, there are further experiments, and one of them, I don't know whether Gerald Schwank is an audience, but they actually took the TNPB that we described and improved it, making mutations both in TNPB protein and guide RNA, and they were able actually to raise the editing activity of TNPB up to 80% in human cells. So that's kind of shows you that TNPB could be improved, but but it's the same question. Why not all of them are functional in human cell?
Okay, if there is no other question, let's...
Yeah,
Oh, no, there is a question over that. Last one.
Thank you. My question is kind of related to the one of Maresca, the first one. I was wondering, so there are autoantibodies that are working fine in bacteria, then are not very efficient in human cells. I was wondering if it's true also from the opposite. So once you evolved your nuclease and it's active in mammalian cells, it's still active in bacteria. Did you test it?
We probably should do this, but when you evolve an enzyme that is active in human cells, you do not care too much about activity in bacteria.
Okay, then let's thank Virgis again for this fantastic presentation.
Thank you.